So I'm Ben Raphael. I'm in the computer science department at Princeton University. It's great to be back at CGSI for another iteration. I look forward to talking to many of you this week. So as we heard already in James' beautiful talk yesterday, the past few years have really seen rapid advances in spatial sequencing technologies, which measure DNA, RNA, or also protein, but I'll focus on DNA and RNA, at locations that are spatially resolved within a tissue slice. So this gives this extra important piece of biological information, the location of the DNA and RNA molecules that we're measuring in the tissue, that's missing from previous bulk sequencing where we just get a single aggregate measurement, or even single cell sequencing where we measure disassociated single cells without knowledge of their location in the tissue. So there are a variety of spatial technologies. It's a really rapidly advancing field. Today I'm going to focus on sequencing-based technologies, so not the imaging or hybridization-based approaches. And even within the sequencing-based approaches, there's many different approaches, and again, with rapid advances. But most of them sort of fall within this framework where we take a thin slice of a tissue, and then at some locations in the tissue, usually these days around thousands of locations, we're able to barcode those locations as we measure the DNA and RNA molecules. And so we can then use standard DNA sequencing to get what looks like type of data that you're familiar with if you've looked at single-cell RNA sequencing data, where for every gene, presumably in the genome, if you're measuring mRNA, you measure the mRNA for all genes in thousands of spots. So you get this transcript count matrix where each entry is the number of transcripts, or really what are called unique molecular identifiers, that are measured for that gene in that spot. But then the new piece is also for every spot, you know its 2D location in the tissue. Now, there are, as I said, multiple such technologies. The one that's probably most widely used now is the commercial technology from 10x Genomics, whose resolution is about 50 microns, which is not single cell resolution. It's about maybe on average 10 cells. But there are other technologies, and I'll show you some data from them as we go, that have close to single cell resolution. Now, I should say, I'm going to talk about RNA and spatial transcriptomics today, but I'll actually show you one brief bit, and there's a lot more going on in measuring spatial DNA, both for measuring genome and for measuring epigenome, such as spatial ATAC-seq. So what can we do with spatial transcriptomics? Well, pretty much anything you're used to doing with single-cell RNA expression, but better, right? Because you have space. So we could cluster spots by their gene expression and find cell types. But now, in addition to finding these cell types that vary by their gene expression, we know the spatial locations of those cell types. So here's just one example of a spatial transcriptomic measurement of the human dorsolateral prefrontal cortex. And this is actually work where they sort of did a manual annotation of these spots into the distinct layers of the cortex. So white matter here in pink, and then multiple layers going through. Here's another example from a tumor. Here's the histology image of the tumor, but now with spatial transcriptomics, we can not just see the histology, but we can see the gene expression signatures that are picking out these different locations within the tumor, find cancerous cells, infiltrating immune cells, et cetera. Looking not just at cell types, but individual genes, we can look at genes and their expression across all spots, but now we can look at the spatial variation in the expression across the tissue. So here is an example of a marker gene for one particular layer, this green layer 3, in this dorsolateral prefrontal cortex. So that's the great news. You know, the somewhat less great news is that the measurement techniques are limited in that we tend to have limited coverage per spot. So just like with single-cell RNA-seq, we have this problem of having, generally low coverage per spot, so a very sparse transcript count matrix. Here, just sort of one example where in this particular experiment, there were per spot on average around about 2,000 transcript counts total for measuring all the roughly 20,000 genes in the human genome, but you'd like to, of course, measure their dynamic expression as well. So lots of zeros in this matrix, lots of sparse data. So how do we deal with sparse data? Well, I mean, if your data is random, then you can't really do anything. But of course, any biological data has structure. And so we can use the structure in the data to help us overcome the problem with sparsity. And so in spatial transcriptomics, I think in the early analyses of this, researchers tended to use what we already knew about uncovering structure in single-cell RNA-seq, so doing various clustering, low-dimensional representations, manifold representations, et cetera. And then after analyzing the transcript data, they layered on some spatial analysis. So looking at once we, are these cell types spatially varying, are the genes spatially varying, and there are various methods in spatial statistics for answering those questions. I just sort of put one here. But then, of course, what we'd really like to do is not analyze spatial at the end, but can we use the spatial information to help us overcome sparsity? So analyze both gene expression and spatial at the same time. And so we can then look at correlations in the data due to space as we're analyzing the transcriptomic information. So for example, we tend to think, and it seems to be true in most tissues, that the gene expression is not random. The distribution of cell types is not random. So for example, cells of different type tend to be near each other, or genes tend to have sort of coherent spatial expression patterns. So we can use models, like here a Markov random field, if we think of cell types, and each of the colors here is a cell type, and we think that maybe neighboring cell types tend to be near each other, and so we can model that through these correlations, or we might use sort of Gaussian processes that model this type of correlation through the covariance matrix of the multivariate Gaussian, or as you saw yesterday, we might use neural network and graph neural network approaches that model the correlations between neighboring spots. So these are a number of the approaches out there, and what I want to show you today is two approaches we've developed to sort of use structure to help overcome sparsity. The first set of approaches is going to be when we just have a single tissue slice, and what we're going to do is try to model these spatial correlations, but really try to model the whole geometry of the tissue. It's a method called Belayer, and a newer method that I'll show you. And then quickly, if there's time at the end, I'll show you another set of methods we developed that take multiple slices from the same tissue and align and integrate them so that we can sort of overcome sparsity in one measurement by sort of pooling them together. And here's just an output of that where we align 16 slices of spatial transcriptomics from a Drosophila embryo. All right, so for the first part, leveraging the spatial organization of the tissue. So as I mentioned, there are many methods built on different techniques that really sort of model spatial organization in a local way, right? So if I'm looking at a spot here in the center in red, then I might look at the neighboring spots, and that might help me say something about what the expression of a particular gene should be correlated with the expression of its neighbors. And so numerous such approaches, I just listed a few, and this slide's probably already grossly out of date. But what we were sort of interested in, looking again at that prefrontal cortex slice, which I'll show you, and I'll show you here again, is that sometimes tissue slices have more of a global structure. And in particular, there are numerous tissues in biology, here layers in the cortex, here layers of the skin, that have this distinct layered structure. And so if you think a bit about these layers, you might think, well, there's maybe genes that determine these layers, and their expression might be sort of constant within the layer. So how could we model this sort of global structure? It's the way we might try to model this. Now, in our field these days, it's sort of, I guess, the popular thing to do is to jump to the most complex model possible. But I'm going to do a more classical approach here and try to do the simplest thing possible, motivated by the simple structure of this slice. If I were to just rotate this cortex slice, so I align it to the x and y axis, then I could think for, say, just a single gene, maybe a gene that's a marker gene for these layers. What's the expression function of this gene as a function of space? What would this function look like? Okay, so if we look at the structure here, and if we think, the first thing we might notice is that by this rotation now at every x coordinate, the expression is the same. Okay, so now our problem is a one-dimensional problem. And then the expression is also constant along particular intervals of the x coordinate until it jumps between these tissue layers. So a function that would describe that is just a piecewise constant function. Okay, so this actually is sort of real data here showing you just the zero pattern of the counts here. And so what you can see actually is already by doing this sort of model, we're able to by thinking that expression is constant along each slice, start to fit this to this kind of piecewise constant structure. Okay? Now, constant expression happens. Certainly it should happen if these layers are the same at every x coordinate, but there are also known to be gradients of expression across space. So if we wanted to take sort of the next simplest model and allow for these spatial gradients, we can instead replace these constants by linear functions so that we get a piecewise linear function of this x coordinate. So we might have a gradient of expression in this gene, and maybe it's high at the start of this layer, and then it decays as it goes towards the next layer. So we can now think of doing a piecewise linear fit of this gene expression. Now, this was a really simple case, right? Everything was lined up, the axis. Of course, that's rarely true even for the tissues that I showed you, although this one is real data. But in general, these layer boundaries are going to be curved. So how might we deal with these curved layer boundaries? And we were looking at this and thinking about it, and we then remembered some math we had learned a while ago. I don't know if anyone learns this math anymore, but there's actually these beautiful maps that will take curved contours like this and turn them into these nice axes-aligned contours, and do this in a way that sort of preserves the local geometry. And in particular, what it preserves is the angles between curves right here. Okay, so this is what's called a conformal map in complex analysis. But actually, I can describe a simpler form of this for you, which is if we think of these curves in this layer as sources of heat, what I'm going to do is assign heat 0 to one boundary, heat 1 to another boundary, and then just ask what's the temperature at each internal location in the layer. And solving that heat equation gives me what are called the isotherms, the curves of constant temperature. And those curves of constant temperature are exactly this coordinate that describes this depth or location within a layer. So we don't need all the fancy stuff, but we do need something from solving the heat equation, or what's called harmonic function. And so we can compute this single coordinate for, in essence, an arbitrarily curved function. So we called this method because of the layers and because one of the authors, a student Chitra, is a rock climber. They call this Belayer. And this is also joint work with Song Ma, who's a postdoc in my group. So the idea here, again, is that we model gene expression as this piecewise linear function of this one-dimensional coordinate. And this one-dimensional coordinate is obtained by modeling this layered structure of the tissue with these arbitrary curved boundaries that form these stacked layers. Okay, so putting it all together, if I just give you data and we want to fit this model, then we have to do two things. If I give you data, my data comes in the form of these transcript counts and with corresponding spatial locations. Then I'm going to find these maps that transform the geometry to this axis-aligned geometry and simultaneously find the piecewise linear functions that maximize the likelihood of observing the data. Okay, and you can use whatever model you want here for modeling the transcript counts. We used a Poisson model to model the transcript counts as a function of this spatial coordinate. Turns out this is a really hard problem to solve. But there are three cases that we can solve that actually tend to be quite useful. The first case is the case where the map is already given to us. And that's the case I already showed you. If your coordinate, this one-dimensional coordinate is already known, if you've already done the transformation, then you're describing expression as a piecewise linear function of this one coordinate. And that's just segmented linear regression. And algorithmically, you can solve that very efficiently. Just a dynamic program, and you can solve it for every gene using the same layer boundaries by running the dynamic program in parallel, if you'd like. Okay, so that's nice. Someone told you already how to align these things together. Another case would be if we just know kind of some approximate layer boundaries, maybe from the start and end of our stacked layers, then we can interpolate the boundaries in between by solving the heat equation. And then once we have those boundaries, we can then run, again, segmented regression. So that's another case we can do. And then the third case we can do is when the boundaries between these layers are lines, but they're not parallel lines, they're just arbitrary non-intersecting lines. And this one we can solve, and maybe for those of you that have taken a bioinformatics class, I don't know if they actually teach in undergrad classes the Nusinoff algorithm for RNA folding. Do they teach this? Anyone learn this? So sad. So someday, if you take a class like that, they may teach you that there's this beautiful algorithm that for RNA secondary structure prediction that deals with finding base pairs in an RNA molecule that don't cross each other. It's called the no pseudoknots condition. It's the exact same condition we have here with our lines, that they don't cross each other. So there's a nice dynamic programming algorithm that's sort of modeled on this same idea where these lines don't cross. You can actually think of like starting at a corner and going around, and this actually looks like one sequence. But if you haven't seen Nusinoff, you won't be impressed by this. So we'll move on. So here's just a quick slide of results just to show you, one, that it works, and two, actually that it outperforms some more complicated approaches. So here's the manual annotation of these cortex. Here's Belayer. You can see it's not perfect. This is the version where we did the arbitrary lines. But if we look at how well we match the manual annotation of the layers, we actually did quite well. We beat fancier stuff like a graph convolutional neural network and a Markov random field type approach. Even on the curved layers, we can do quite well. And what we also can do then is get these gradients of expression, something that these other methods don't often produce. So we can see actually genes, known marker genes for certain layers in some cases, novel candidates for marker genes that have increasing expression in this case or decreasing expression in this case as a function of their location, their coordinate inside that layer. Okay? So we have a number of such interesting cases like this, and there's a lot more in the paper. This is published work, so you can go look at the paper. We've recently extended this beyond spatial transcriptomics as some newer technologies, spatial multiomics technologies. Raphael, who's at Yale, happened to be visiting one time and just started showing me this beautiful data he had from his spatial cut and tag and RNA combined, so spatial multiomics. And he's showing me this data, and I said, oh my gosh, let me show you this method we have for finding layered structures. And he said, wow, this is incredible. Can you analyze our data? And we do. And what we can sort of see is that we can pick out these layers quite beautifully from RNA, from, in this case, epigenetic mark of histone 3 K4 methylation. And in fact, if you overlay them in the multiomics, the layers are quite concordant between these two modalities, which is quite nice. All right, so that's Belayer. That's the sort of published work already. It's this global model of spatial expression with these layered tissues. You simultaneously infer the layer boundaries and you infer these piecewise linear expression functions. And you can pick out, I didn't really show you the metrics here, but you can pick out interesting genes by genes that have either discontinuities in their expression as you jump between layers, or that have gradients of expression. So pretty much anything with a non-constant slope as you look over your layer becomes a candidate for an interesting marker gene. And you can actually find, with pretty high fidelity, many known marker genes and get some novel candidates, as I showed you. All right, well, we're very proud of Belayer, and it involves some beautiful mathematics, harmonic functions, the heat equations, but at the same time, learning all this mathematics is a bit of a chore. Who has time for it these days, right? So, following sort of the current trends in science and society, we're going to replace the sort of natural intelligence approach by artificial intelligence, right? Specifically, like, deep neural networks, which actually are already kind of passé. So, we'll use an LLM next time, I promise, But here's the neural network. So this is a work of Gustav, who just suggested, hey, why don't we just try to use a neural network to learn this mapping, this function. Neural networks are really good at learning these functions. So we try to predict gene expression from its two-dimensional coordinate, but processed through this one-dimensional coordinate. And I thought, yeah, sure, go try it, And the thing just worked, and it worked on like much more complicated tissue geometries than we could analyze with the sort of complicated stuff. So here's a mouse cerebellum, and you can kind of see the different layers that we're picking on. But then the other thing was that we said, why is this working and what's going on and what else can we do with it? And we sort of realized that another thing we could do with it was actually just pick out gradients of expression and find these spatial gradients, which is sort of embedded in this 1D coordinate. So let me show you a little bit how that works. And here I'll go to a calculus interlude. Some of you are like, gosh, he showed the neural network for one slide and now he's back to the math. I was like, what a downer, Yeah, but no one ever looks at it. It's just run the PyTorch package. But here's the calculus. So this function, this expression function, is a function from location 2D to RG, right, for every gene, so high dimensional thing. Let's assume that we have these different regions, right? So there's different regions or layers somewhere. We don't have to prescribe where they are. So this function we want to learn is going to be continuous, but actually it's important to allow these discontinuities at these layer boundaries because they're very different organization of tissues, as cell types have discontinuous expression as you go from one to the other. So, we want this piecewise continuous function over this region. Now, as you recall from calculus, at every location for a function like this, you can compute the gradient of each coordinate, right? Here's the gradient of say, the first gene, it's got a gradient at every different location. And this gives you what's called a vector field, right? If you look at all these sort of vectors together. Okay, so this is sort of for one gene. But then, if I look at other genes, there's one point gradients might be in different directions, right? So that's fine, that's just calculus, and you can compute all these gradients if you knew this function. Of course, we don't fully know this function, right? It's very sparse data, that's part of the problem. So, does the tissue organization impose some constraints on this vector field? And with some mild assumptions, we can actually sort of see that for genes that are sort of variable genes, marker genes, or if you want to do this with sort of principal components, sort of assume that their gradient directions are going to line up within that particular location at the point. And then the gradients all together we can describe, one way to describe a vector field is as the gradient of this function called the potential function. Okay. Now if you took any physics, I won't ask if people took physics, so disappointed in the Newsonoff thing. If you took any physics, you'll remember that there's these potential functions. Heat is a version of potential, electrical potential. So you've got these curves of constant potential, and then the gradients tell you the direction of maximum increase. Right? Well, if you don't like that assumption, these things, of course, come up all the time. If you're going to go hiking, here's the Hollywood Hills. If you're going to go hiking, you might be used to seeing a topographic map. Here's a map. That's really a potential function for height. Here's again for weather, the isotherms of heat, right? So here's sort of this potential function. And so following this terminology, we're going to call this coordinate for our tissue slice the isodepth. These curves of constant coordinate, right? Location within our tissue. So this is sort of a generalization of what we had before. And then we can find the gene expression function for a given gene as a function of this one magical coordinate, the isodepth. Okay, so we get the topography of the tissue slice. All right, so putting this all together is a new approach, which we backronymed Gaston. Ustav, who's a rock climber, tells me a Gaston is a move in rock climbing. I don't know. Some of you that are Disney fans might have another word association with Gaston, which I thought is what he was talking about, but it's not this. So, the idea again, spatial transcriptomics data. We just throw it into this neural net, but because of the way we set up the neural net, we have this interpretable coordinate, right? So this is interpretable deep learning we're doing for those that are keeping track of this stuff. And we get these expression gradients. We can find these, it says clustering, but really the layers come directly out of the method. And then we can look as a function of now this isodepth, which is much more general than what I was showing you before, expression as a function of that depth. And we can look at changes in gene expression. We can look at changes in cell type proportion. All right, so let me show you some examples. Here's that mouse cerebellum data set. This is mouse cerebellum. It's through a technology called SlideSeq. It's V2 of SlideSeq. It's actually very low coverage per spot, so it's very sparse data. But what you first learn is this isodepth. You learn the vector field. Here's the corresponding layers. They actually look quite good. You compare them to what you got with a graph convolutional neural network. It's a little bit cleaner. The spatial coherence is much higher for our method than it was for the GCN. GCN uses only local information. And then just, here's a comparison of the cell types. So RCTD, for those of you who don't know, this is a method that uses a single cell RNA-seq atlas to layer on top the cell types from the single cell RNA-seq onto the spatial. So this is sort of much noisier because it's showing you potentially where every single spot is, and the cell types and their spatial locations. But if we sort of use that, those cell types as marker genes, we can see that we have higher spatial coherence while getting kind of similar annotation of the dominant cell type in each layer as the convolutional neural network. Let me show you that as a function of the 1D coordinate, which none of these methods have. So then now, having collapsed all of these layers, like everything in this green has a depth coordinate, everything here, we can watch as we go along from outside in, from pink to green to et cetera, pooling information across all the spots from the same iso depth in the layer. We can plot the gene expression. What I'm showing you here is the changes in annotated cell type as a function of this coordinate. So this pink layer is dominated by oligodendrocyte cells. And then we have a rapid shift right here to granule cells, which is the granule layer. And then right here, interestingly, this is the Purkinje. I'm mispronouncing that. Purkinje-Bergman layer, which actually, interestingly, people call this a single layer, but what we can really pick out is these initial one type of neuron, one cell type, the Purkinje neurons, and then they rapidly sort of decay, and then you get the Bergman neurons coming up, all within this one sort of layer. There's actually just sort of a small overlap between these two that's been recorded in the neuroanatomy literature. So this is sort of interesting for cell types, but then we can look at individual genes and their expression, and we can pick out marker genes as a function of this isodepth, look for genes that have either discontinuous expression, as shown back here, genes that have changes in their gradients as you go along this coordinate. Those indicate potential layer-specific genes, and we do much better in terms of picking out known marker genes compared to numerous other methods that have been developed in the spatial transcriptomics world. Also, by combining both of these things, we can look at, for individual marker genes, is their change in expression due to just a change in the composition of the cell type, or is it due to some other gradient in expression for that gene? And so we can separate out these genes with expression gradients into ones that are independent of the cell type, because they change even though the oligodendrocyte in this case is changing in the same way, and ones that once you zero out the cell type, although there's a change right here, it's really just a function of the change in proportion of the cell type. So this is another sort of nice feature of the approach. Here's just one more example to show you another kind of geometry. This is the olfactory bulb from mouse. This is another spatial transcriptomics technology called StereoSeq. In many spots, really, really low coverage, but very high resolution. So we can find the layers. Here's the isodepth, these gradients pointing from outside in. There's another method, spaceflow, that doesn't quite sort of get the layering structure right. Here's Gaston. It gets sort of all the layers quite well, including the center layer where there's sort of stem cells, seemingly better than the graph convolutional neural network. Again, we can look at cell types and we can look at individual genes as a function of this isodepth and see gradients of expression as you go between the layers of the olfactory bulb. Okay, so that's a summary of Gaston, these spatial gradients across the tissue slice, this isodepth giving this global model of tissue organization. It's via this neural net that learns this just directly from the transcript counts. And then you can look at cell type driven and cell type independent expression as a function of this layer depth. So this is new work, preprint coming soon. Are we posting these talks on YouTube? Are they coming out on YouTube? Or? Yeah, but in a while. Okay, so it's fine. So for the YouTube audience, I'll give the pitch. Look below for the link to the preprint. And what do you think about spatial expression? Let us know in the comments. Subscribe to our channel. There you go. Just increased your ad revenue. My kids all the time. That's all I hear is the YouTube, right? Yeah, I know, right? Yeah. Well, CGSI will get 3 cents. So in the last few minutes, let me show you a quick version of the multiple slices. This was a method actually that I described originally at CGSI last year, but we have some new work that I wanted to quickly highlight. So the idea here is you've got multiple spatial transcriptomic slices from the same tissue. Can you stack them up and align them in one mode to sort of get pairwise alignment? Or can you take all the slices and sort of integrate them into one center slice to sort of help overcome sparsity by pooling information from multiple slices? So this was paced. It came out a little over a year ago, and we were very excited to use it, and we have been using it. But we discovered sort of one issue with the algorithm, and that's that in many real applications, the slices may overlap only partially. And this is just sometimes a technical artifact. This was data from our collaborator, as liver metastasis slices, 2 slices from the same liver met. And just the way they put them on the array, they just don't overlap the whole region. They only kind of overlap in the center, in this red highlighted box. Or sometimes you're just aligning slices that, even though they're consecutive or nearby, the tissue composition is slightly different, right? So when we did this Drosophila embryo, the tissue composition changes. Actually, just that I show you that embryo again, just because it's sort of an ellipsoid, the size of the layers changes as you go from top to bottom, right? If you cut it this way. So, long and short, paste is sort of doing a global alignment, if you will, trying to align the whole slice. And what we want is kind of partial alignment. So, near Hollywood, so we have a sequel, right? A sequel, paste 2, even better, paste 2, partial alignment of these slices. So, again, multiple slices from the same tissue, but now instead of trying to align them globally, you try to find the best portion to align, and then you can stack them up in 3D. And we can sort of estimate what fraction should overlap, and we can reconstruct in 3D. This is work of Jinhao Liu, a current PhD student working with Ron Zeira as a former postdoc. I'm not going to go into all the details. This is also as a preprint. This was at Recom this year. Genome research version is coming out momentarily in a few weeks, and so you can read more of the details there. I'll just quickly say something about the methods that, what this operates on is a beautiful framework called optimal transport. So optimal transport is a way of generically mapping a probability distribution to another probability distribution, but while minimizing the amount of work you need to do to move from one distribution to the other. So sort of classically, if you think of this these probability distributions as like a mound of dirt, you want to transform this mound of dirt to this mound of dirt, but do it with moving as little dirt as possible to minimize your work. Optimal transport gives you a way of doing that. So all the flexibilities in how you define this work, the so-called transport plan. So what we can do is define a transport plan to talk about moving spots to spots, in fact, moving them probabilistically, so probabilistic mapping, so every spot could have some fractional mapping to every other spot, but doing this in a way to minimize the transcriptomic distance between map spots and minimize how much in space you have to move the spots. Okay, so that's what's sort of happening here. You're just sort of trying to move one slice to another probabilistically according to both spatial similarity and transcriptomic similarity. And in PACE 2, because we want the partial alignments, it becomes what's called partial optimal transport. Right? So you don't move the whole mound of dirt, you move part of it. Right? There's a nice framework for this. No, not quite. So, yeah, not quite. We can talk about that. They're similar but slightly different. So here's just the example of the Drosophila embryo. I showed you this on the first slide. You can kind of see this ellipsoid shape I was talking about. Here's the slices as they were in 2D, so slice 1 to 16, right? So the small slice on the bottom, I guess, here, going through to the middle. And this is just some comparison showing that, paste 2 in blue, if you sort of look at how accurately did you map the previously annotated cell types, we seem to be doing quite well, especially in cases where the slices are different in size. So that's the advantage of the partial alignment. All right. Well, wow. I talked fast this time, which is great, because then I can show you the very last bit. So the very last piece here is now that we have paste, we could start to look at another application, which is an application of great interest in my group, which is tumor evolution, looking at how tumors evolve. Now, for a long time in my group and many others, we've been looking at how tumors evolve over time, and tumors evolve over time by accumulating somatic mutations. And so if you measure mutations at the present day, you can try to sort of reconstruct the past using phylogenetic techniques. But that evolution is happening in a spatial context. So we've been very interested in trying to see where are these different tumor clones with different mutations migrating in the tumor. What's the clonal structure of the tumor using both this sort of evolutionary process and this spatial process. Now, the bad news is that there's not yet, there's a little bit of spatial DNA sequencing, but most of the action's been in spatial RNA sequencing. So we get a lot of spatial RNA, not a lot of spatial DNA. And here we're interested in, for somatic mutations, measuring genomic changes. Now, you can actually try to get out some genomic changes by looking at RNA. And in particular, you can actually get at these larger changes, so-called copy number aberrations, where big regions of the genome are duplicated and deleted. And the reason why is that if you have a big duplication or deletion in the genome, it changes the expression, or can change the expression, of many genes that are in that region of the genome. Right? If I delete a whole chromosome, then all the expression of those genes in that chromosome are going to be perturbed. Right? Anything that's normally expressed then suddenly would be less expressed if there's one fewer chromosome. If you delete both chromosomes, nothing from that chromosome can be expressed. Right? With amplification, there's also some increase in expression that can happen. So large copy number aberrations alter the expression. So you could try to identify copy number aberrations from transcriptomic sequencing data. People have tried this from single-cell RNA-seq with some success. However, there's lots of changes in expression, as we all know, that have nothing to do with copy number changes. Most of the changes in gene expression have nothing to do with a copy number change, right? They're just gene regulatory processes. So we sort of thought, using the spatial information, we could perhaps get a better view of the copy number aberrations, because we could also sort of assume that genetically similar clones, genetically similar cells, are likely to be close to each other in space. Okay? So there's another signal, not just finding these copy number profiles across the genome, but also these copy number profiles correspond to clones, as they're sometimes called, and similar copy number profiles should label spatially close regions in the tissue. So this is a second signal. Adjacent spots should be genetically similar. And a couple of years ago, we developed a method called STARCH to identify copy number aberrations from spatial transcriptomics data, work of a former PhD student, a former postdoc, and I actually described this a little while ago at CGSI. And I think I pulled the slide because I didn't know I'd talk so fast. But the way we sort of do this is we just model two types of correlation, right? Along the genome, there's correlation in expression due to copy number changes. And we can model that with we use a hidden Markov model. There's different ways you could model this correlation across the genome. And then the correlation between these clones is correlation in space. As I told you, there's different ways you can model correlation in space. Here we used a Markov random field. So we have a Markov random field, and then we just have these coupled together. So there's a Markov hidden Markov model and a Markov random field that you infer jointly. And that's what happens in STARCH. Song Ma and my group has extended this. I'm not going to describe the extension into a new approach, Calico ST, that has some really beautiful things, which I'll skip. But I'll just show you one piece of combining these two algorithms where we have multiple slices. These liver mets I showed you, 2 liver met slices. We align them with paste. We then can build this adjacency graph in 3D now, and then run the new thing, Calico ST, to find these copy number aberrations and clone labels all together. And what we pick out are, here's regions of normal cells, 3 distinct tumor clones in this liver met, distinguished by different copy number aberrations across the genome, shown down here. And you can kind of start to see, and actually I didn't show it on this slide, but we can pretty confidently relate which clones are descended from which ones by looking at the copy number profiles, so we can start to pick out the phylogenetic tree evolving in space. I cut that off the slide, so I apologize for that. This is also coming soon, so you'll see Calico ST very soon. So here's just a summary of this last quick part. So paste 2, the sequel to paste, partial alignment of these multiple slices, and then once you have that alignment, you can start doing lots of cool stuff with it. You can look at anything you look at in spatial transcriptomics, but now in 3D. spatially varying genes, cell type composition. I showed you one application with copy number aberrations in cancer. So I think, hopefully in this talk, and James's talk yesterday, we've really shown you that, these spatial technologies are really advancing at a rapid clip. Many different modalities. I mentioned spatial attack, spatial DNA, newer technologies that couple imaging and sequencing. 10x Genomics has Visium. And each of these just has a bunch of new computational statistical challenges. So I really think a ripe field for students and postdocs to find great projects. And I think there'll be many more talks at CGSI to come on really cool things happening with applications of spatial transcriptomics through novel methodology. So with that, I'll conclude. Just thank the members of the group in bold who worked on the projects I discussed, and many other members of the group who've contributed and helped us make these better. Funding agencies and the software for all but the new stuff is there, and feel free to reach out. If you don't find me at CGSI and you have other questions, feel free to contact me. So thank you very much. Happy to take questions.